Okay, let's have a class. Um, we will talk about the poultry meat microbial safety during the processing today. But before we talk about today's class, let's do some of the review for our last section. Something of key importance I want to mention is you can review because you're going to be in the final exam. So the first thing very important, when we talk about hazard, if you remember, what's that? Hazard analysis and critical control points. Okay, now why this one will be happening? Remember, the Hamburg killed kids in 1994. So 1996, there's a federal register which is talking about pathogen reduction and hazard plan. And hazard, HACCP. And that requires all the meat and the poultry processing plants need to establish a hazard plan. And I mentioned about the detail of the hazard plan. That's another class, which is hazard class or a food microbiology class. And the certification what we can get is from after training you develop a hazard plan, which is from Texas AEM University, which is called hazard Alliance certification. Now Pennsylvania State University Extension Service can have some type of training. Now here what we need to know, what is the 7 principles of hazard? Number 1 is conduct hazard analysis. That including physical, biological, and chemical. Number 2, establish a critical control point, which is CCP. Number 3, establish a critical limits. I just say CL. Now, critical limits, critical control points is any step during the food processing which is control can be applied to reduce food safety hazards below the regulation or eliminate that. Now, every CCP is corresponding which is to critical limits. The critical limits is a physical or chemical parameters corresponding to that, and this should never be is a range. Okay, when we have this, we have to establish a monitoring procedure. Because we need to make sure the CCP and the critical limits, both CL, also works. And number 5, we need to establish corrective actions. So if anything happens, we call it a deviation happens, we need to take corrective actions. Corrective actions. Number 6, we have to do a validation, which most of the time is in-house, and the verification procedure. Verification, which means the whole system has to do a verification, usually by a third party. And the last one, very important, is record keeping and documentation. And this is a key. Every data log, every record, you need to have it. But make sure hazard for what we talk about. This is a proactive food safety approach. But do not make assumption it's guaranteed food safety. It's not. It's a proactive food safety approach. Okay? But not guaranteed food safety. Based on this, we need to know the other things. This hazard plan, we also mentioned briefly maybe. There is a 2 prerequisite program you need to have it. Prerequisite program. One of the two prerequisite program is GMP, good manufacturing practice, and SSOPs, sanitation standard operation procedures. These things you must be established first. Then we're going to talk about is a HACCP. Okay? Now other things, we mentioned about the fresh produce. Fresh produce, they do not have a HACCP. Juice, even some dairy products and meat, they all have poultry meats, red meats, they all have the process, have some module for HACCP. Fresh produce, they don't have it. So what we have to do? We have other things, which is GAP. What is GAP? Good agricultural practice. Now, all the key points which they talk about. Number one is safe soil. Number two is clean water. Safe soil, which means the soil you're using, no harmful bacteria, especially as they relate to the manure and the compost. And the clean water, there's no coliform and the fecal coliform. And what else we have is clean surface. Clean surface we talk about is cleaning and sanitation and sanitizing to eliminate typically biofilm control. And the last one is clean hands. And this one we talk about these days all the time, which is wash hands using antimicrobial soap for at least 25 to 30 seconds. Now, how you verify that? Sing a happy birthday song. Okay, that's important. These are the things. Now, related to that, the federal regulation is FSMA, Food Safety Modernization Act. Okay, this is really flexible for small local growers, and they give some exception for it. Now, FSMA signed into the law, which is January 2011, for the Safety Modernization Act. Now, then we talk about the passages. Remember several passages we talked about. Today we're going to talk about some new, we're going to mention some passages. We talk about this E. coli, 0157H7. This guy you need to know, number one, this is zero tolerance in, I will just say, non-impact beef, including ground beef. And why does this have to be zero tolerance? Because it has a Shigei toxin. And the Shigei toxin, what we talk about is STX1 and the Shigei toxin 2. Now, more important is this too, for toxin and also for rapid testing. So there is two things to talk about, is toxin and also for rapid testing. Now, Shigei toxin, why it is a risk? Because it will be attacking CB3 in kidney. Once they attack it, it becomes an AV toxin, will cause a very severe symptoms. We call it HUS. Hemolytic, uremic syndrome. Okay, water diarrhea with blood kidney. Could be attacking the neurological system. Will be dying if kids get contaminated. Okay. Now we also mentioned another one is Listeria monocytogenes. Listeria monocytogenes is a risk which is ready to eat meat, like hot dogs, turkey breasts, ham, because there's outbreak. Remember the outbreaks. Multi-state outbreaks happened in 2000, early 2000. Okay, now what is the standard? What is the standards for Listeria monocytogenes? It is not zero tolerance. Less than 1 per 20 gram meat. Okay? And I said it's an emerging pathogen in fresh produce and also in the poultry meats. Listeria monocytogenes is called steel baby, miscarriage, and also called meningitis. And then, how are we going to prevent it? Remember, USDA, FSIS, and the FDA have 3 alternatives that is used to control the Listeria monocytogenes, including that's called post-mortality treatments, okay, with antimicrobials. Then all antimicrobials. This is the second one. Third one, which is sanitation, sanitizing plan, and the sampling plan. So these are the things we do, some of the conclusions before we move forward. Okay? So today what we're going to do, today we're going to focus on is pathogen control during poultry processing. There is a lot we could talk, and it's very interesting, some of the topics. So we want to mention these one by one. Especially during the processing. Lots of the stuff we can cover, it's related to the microbiology, and the applied microbiology is very, very interesting. So we want to talk about these one by one. First of all, you're going to ask me, how you come out, you're going to talk about poultry processing. And I'm going to show you here. This is the broiler production by state per million head in 2014 by USDA do a survey. One of the big poultry states in the United States, you should know, is in the South. Where are they? Georgia, Alabama, Mississippi, Arkansas, Texas, North Carolina. These are the big poultry states. So, in other words, the poultry production is somehow at least more than 500 million head per year. And all those places, the land-grant university has a big poultry science department. Let's say Mississippi State University have a poultry science department. Auburn University in Alabama has, University of Georgia has, Texas AEM has, North Carolina State University has, because it's a big market and a big industry requirements there. So if you're interested to study poultry science in a graduate school, then you should find those places, because there's a market there. Then we look at that. You see, Kentucky a little bit, 300 million heads. I used to work at Kentucky. Now they did not develop very well because the poultry litter sample and the waste is very difficult to control. So they try to remove it. When you look at here, did you find West Virginia? And West Virginia history is mining, little bit steering, and the rural medical health, and the mountain area. Is that right? There's no big industry. There's no big agriculture industry. We talk about fresh produce, they are all farmers markets, small. But you look at the poultry, how many are there? 95.3. This is ranked almost about 13 or 14 among the 50 states in the United States. So I will say the boro industry in the West Virginia is a major part of the agriculture. Now we do not have like a large company. Remember Tyson, Smithfield, all those companies, they are in Arkansas, they are in Texas, Alabama, Georgia, all those places. West Virginia, most of them also are small growers. Most of them are in Hardin County, which is on the east side, close to Virginia and Maryland region, across the state border. Okay, just across the state border. So just want to let you know. Boro production in West Virginia is important. That's why I want to have this section talk about it. Okay, this is a microbiology class, so we care about passaging. Now, what are the important passages for the poultry meats? All these are important, but what are the most important? Campylobacter and Salmonella. Those are the two are most important. Now, people are going to talk about E. coli. Well, maybe, Listeria, maybe, Clostridium happens, but more important for the meat, broiler growers, Campylobacter and Salmonella are the two most important. Now, we're going to mention a little bit about that. Okay, let's talk Salmonella first. What is Salmonella? This is gram-negative, large shape, facultative bacteria. And we know that, and it's a mesophile. And I'm going to mention this mesophile is important. Later on, we will talk about it. Okay. Then what is Campylobacter? Campylobacter is also gram-negative. It's like a curved shape. It's not a facultative. It is a aerophilic bacteria. This is the key. What that means, they grow better with 2 to 5% oxygen. More than that, Campylobacter will be down. It's a key. So Campylobacter is very interesting. It's very ironic. This pathogen is not going to grow very well, especially very difficult to grow in the lab situation. However, it's a risk of the poultry meats. So people will say, is that be so dummy if you have a poultry meat just exposed to the oxygen and they died? No, not really. They survive very well on the poultry, in the intestinal, in the C-car, and also on the skin surface. And I'll show you about that. Okay, so these are the two things I really want to mention. Then we go back to look at the slides. This is Campylobacter. Curved, motile, large, gram-negative, and relatively fragile, sensitive to environmental stress. Now this is a key, requires 3 to 5% oxygen, 2 to 10% carbon dioxide, and for optimal growth condition. Okay, you can see that it can survive in a wide variety of the wild domestic animals. The birds can have it, chicken can have it, turkey breast can have it. 2 to 4 weeks grow, as low as 40 F. What that means? Like the refrigerator temperature, they can survive there. At the room temperature, 2 to 5 months. What is the infection dose? The infection dose is high. It's 500. I would say more than 500 cells will cause problem. Okay? Now let's talk about Salmonella. This is the one you know about that. Log-shaped motile, non-endospore forming gram-negative, grow well in the body temperatures. That's why we call it a mesophile. Somewhere between 30 to 45 degrees Celsius, they can all grow very well. And at 40 degrees Celsius, they grow much better. And then what we have, that can survive in a very wide range of the pH, as low as 4.0 and higher than 9.0. And the optimal pH is between 6.5 and 7.5. And grow very well, 35, 45. Can be grow as low as 4 degrees, 5 degrees Celsius, maximum growth 45, 47. This is the key I want to mention. This guy, the temperature for Campylobacter, 42.5 is the maximum optimal temperature you can grow. For Salmonella, basically it's 35 degrees Celsius. However, 45 degrees Celsius, they also can grow. This is the key. I will mention the process. It's important. Okay, this is some surveillance serotype in 2005. Based on the CDC data, one of the #1 cause problem of the Salmonella is Salmonella typhimurium. Responsible almost 20% of all the cases in 2005. Second one followed is enteritis. Salmonella enteritis is a very typical pathogen on eggs. I'm not sure you looked at the news. Last year, there's a big outbreak or recall happened in Indiana. It's an egg company. They are isolated with Salmonella enteritis. So the second one is actually more popular than the first one. Newport is another one, and Hindenburg is the other one. These four, almost you can add up them together, almost about 50% of all the serotypes outbreaks happened in 2005, and it's pretty consistent year by year. So when we do research, we care about Salmonella typhimurium. We get isolates from Salmonella enteritis, have Newports and Hindenburg. Those will be the targets, serotypes for Salmonella in the poultry processing. Okay, now we want to talk about something. Why didn't remove here? Okay, sorry. E. coli 0157H7 in ground beef. Because of that outbreak, a big outbreak, it is zero tolerance. You're not allowed to have one cell of E. coli 0157H7 in a ground beef. And I mentioned if you have a ground beef, have tested one cell E. coli 0157H7, what do you have to do? Cook to ready to eat meat. Ground beef contaminated become a sausage. Said very simple. Listeria monocytogenes, pretty streak. Can you see it? One cell, less than 25 grams. And that's very streak. Not many. Basically, one hot dog, how many? What's the weight? 50 grams. Only allow 2 cells. How about Salmonella and the Campylobacter? They allow, the standards is pretty flexible. Okay, and I'll tell you what, this is brand new, not really too old. So the new performance standards in 2015, this is FSIS, Food Safety Inspection Service. The new standard performance of Salmonella and the Campylobacter on a carcass. Salmonella is allowed to have 7.6% positive. And the Campylobacter is allowed to have 10.4% positive rate. What that means, you had 100 poultry carcasses, 7.5. So you had 100 carcasses. I'm going to do a sampling. You are allowed to have almost 7 to 8 sample could be Salmonella positive. You are allowed to have 10 of them is Campylobacter positive. This is pretty flexible in a broader carcasses. Now why? We're going to mention about that because there's a multiple location during the processing. It's easy to happen is cross-contamination of Salmonella and the Campylobacter. And you have to control very well. If you didn't control very well, it will more or less have some. Okay, so that's a tricky thing. That's the reason when you go to a restaurant, you order a beef, you will say medium rare sometimes, cooked to internal temperature 65 degrees Celsius. You want to tender this. When you order a chicken steaks, you never say, hey, I want a rare or a medium rare chicken steaks. Never say that. Because there is a Salmonella there is a Campylobacter there, which is expected. Okay, so just want to let you know, the performance standards for seminar and the capital battery is pretty flexible and very low, I will tell you. Now, then what are the industry scale food broader processing? Okay, basically, you had a breadstick. The breadstick ham. Okay, immobilization, we do first of all called exaposition. We cut it, we kill, let it bleed it. Then we do a scalding. Scalding, what that means, I have a tank with hot water, and I put in, let them immerse a little bit, then the feather becomes soft, it's easy to take it off. Then we do pickling, which is hanging there, and take out of the feather. Then we do evisceration, taking out of the intestinal, the C-cup, take it out. All the intestinal structure, take it out, or left the carcasses, because that's the meat what we want. Then finally, we do a chilling. Immersed chilling, we put in a chilling tank. The chilling tank have cold water. Okay, chilling about an hour. And then we do aging. We maybe let them stay in the refrigerator temperature for a week, because we want the muscle to be a little bit tough. Okay, because chicken muscle is pretty soft. We want them a little bit tough. Then further processing, maybe vacuum package, then we go to the market. Now, during this process, I will say during scalding, pickling, evisceration, even chilling, there is those 4 steps right in the middle is a major step, could cause cross-contamination. And I tell you one by one. First of all is scalding. Okay, scalding happened is 1st century AD. You look at the picture there. They do scalding. They don't know the scientific behind that, but they know when you dip a chicken, a hot chicken with feather into a hot water, it's soft, it's easy to take it. This is the industry scale, what they did there. However, the scalding temperature control is very important. Okay, so what are we going to talk? Scalding process is temperature control. There are basically 2 types of the scalding we're going to do. Number 1 is hard scalding. Number 2 is soft. Now, what's the temperature? You can see the hard is somewhere 59 to 64. The soft is a little bit lower, is 51 to 54. Okay, there are the 2 things you never have to do. You never should do. Most of the small processor or small company, when they do scalding, what they do? They're manually doing that. Is that right? This is too hot. Okay, so how do you do it? They are doing the temperature is like 45 degrees Celsius. Warm water. This is wrong. Why?